Plant conservation refers to a broad range of activities aimed at preventing plant species from becoming endangered and ensuring the sustainability of plant genetic resources. Biotechnological tools such as in vitro culture, cryopreservation, and molecular marker play an important role in plant conservation. These tools help scientists study plant diversity, preserve endangered species, and manage genetic resources more effectively. However, one major challenge in plant conservation is the limited availability of genomic resources, which can reduce the effectiveness of conservation strategies and their sustainable use. To overcome this problem, genomic techniques are applied in species and population conservation to obtain critical information on the relationship between genetic variation and the environment. Techniques such as genome sequencing, genome skimming, and genotyping by sequencing, together with molecular markers like SSR, SNP, and chloroplast DNA markers, are used to assess genetic diversity and support conservation decision-making. Biotechnology provides strong scientific support for biodiversity conservation by improving our understanding of plant genetics and helping to ensure the long-term survival of plant species.
For this slide, I will explain molecular markers. Molecular markers are short DNA sequences that are used to detect genetic variation among individuals or populations. They are very important in plant conservation because they help us understand the genetic health of species. Two commonly used molecular markers are SSRS and SNPS. SSRS or microsatellites are highly polymorphic and are widely used in population studies to analyze genetic diversity and gene flow. On the other hand, SNPS are single base pair variation in DNA and are considered the modern standard for high-resolution genetic mapping. Overall, molecular markers provide essential genetic information that support effective conservation planning.
Now, we move to advanced genomic technique. First, biomarker whole genome analysis. In the past, plant genetic studies used only small DNA fragments such as microsatellite and barcodes, which gave limited information. Today, whole genome analysis allows scientists to study all the DNA in a plant. It helping us better understand genetic diversity, inbreeding level, environmental adaptation, and population structure. Second, next generation sequencing or NGS. NGS can sequence DNA quickly at a low yield caused by reading millions of fragments at once. This is especially useful for endangered plants because it helps measure genetic diversity, detect harmful mutation, and support conservation and breeding programs. Last, we move to environmental DNA or eDNA. Plant leave DNA in soil and water through roots, pollen, and decaying tissue. By analyzing this sample, scientists can detect rare or hidden plants, monitor biodiversity, and study ecosystem without disturbing natural habitat, which is ideal for endangered or hard-to-find species.
This slide explains about application of genetic diversity assessment in plant conservation. Assessment of genetic variation can indicate whether plant populations are at low or high risk. Genetic variation reflects the ability of a population to adapt and survive in response to shifting environment. Studies reported in UWI-ST Austin Journal show that populations with higher genetic variation are generally more resilient to environmental changes. And then we go to identifying at-risk population, which is when a population has low genetic diversity. When genetic diversity is low, the population cannot adapt well to environmental change, so the risk of extinction becomes higher. Because of this, conservation uses genetic information to decide which population need to protect first, especially small or isolated population as discussed in BMC Plant Biology in 2024. After that, we move to inbreeding detection. Genetic diversity studies help scientists detect inbreeding depression at the molecular level, especially in small populations. This is important because it helps improve breeding program and support the preservation in increase of genetic diversity. This discussion supported by Mondini in 2009 and Al-Haswani 2024. And finally, we look at a visual example. This image shows the average genetic diversity of different population groups. It clearly compares population with high genetic diversity and population with low genetic diversity.
Okay, let's move to the application taxonomy and species identification. Taxonomy is the science of classifying organisms based on shared traits in genetics. Essentially, it's a way for scientists to organize all living things into hierarchical groups such as kingdom, phylum, class, and species, so that we can understand their relationship and evolution. Species identification, on the other hand, is the process of determining exactly which species an organism belongs to. Traditionally, this was done using morphology like leaf shape, flower color, or other visible traits. But now, we also use molecular techniques like DNA to be more accurate, especially when species look very similar to each other. One of the most powerful tools for species identification today is DNA barcoding. DNA barcoding uses short, standardized sequences of DNA to identify species. Think of it like a genetic fingerprint. This method is particularly useful for species that are hard to tell apart just by looking at them. For example, in bamboo species, many different species can appear almost identical morphologically, but DNA barcoding can clearly distinguish them by comparing their genetic sequences. DNA barcoding is not just useful for research, it also has important applications in conservation. For example, it can help identify protected plant species that are being traded illegally, whether in timber market or herbal medicine products. By accurately identifying these species, authorities can enforce conservation law more effectively and help protect threatened or endangered species from exploitation. Here, we have a visual representation of DNA barcoding. Each color bar represents a short DNA sequence, which acts as a unique genetic signature for a species. By comparing this barcode with reference databases, scientists can accurately identify the species, even from small or processed samples. This simple tool has become a critical part of modern taxonomy and biodiversity conservation, helping us to both understand and protect the natural world.
This slide shows the application of genetic management in plant conservation, focusing on ex situ and in situ management. First, on the left side, we have ex situ management, which refers to conservation outside the natural habitat. In this approach, seeds are collected from wild plant population and stored in facilities such as seed bank, in vitro collection, and field gene bank. The main aim is to store seeds that represent the full genetic diversity of a species, which is important for long-term conservation as highlighted by FAO 2024. This method helps protect plant genetic resources from threats such as habitat loss, climate change, and possible extinction. In this diagram show different ex situ methods, including conventional seed storage, in vitro conservation, and field gene bank, which are commonly used in plant conservation. On the right side, we have in situ management, which focuses on conserving plants within their natural habitat. In this method, the best source plants are selected for replanting and genetically diverse, locally adapted seeds are used. This approach helps improve the survival and resilience of replanted forests, especially under local climate change conditions, as reported by Thomas 2014. This diagram shows in situ conservation, which means protecting plants in their natural environment. At the top, on-farm conservation refers to plants conserved in farm field, local population, and tribal area. Farmers continue growing traditional crops, helping maintain genetic diversity and local adaptation. At the bottom, natural habitat conservation includes biosphere reserve, natural park, heritage site, and sanctuaries where plants are protected in the wild with minimal human disturbance.
Now, we move to impact of biotechnological tools. Firstly, in terms of appreciation, biotechnological tools such as molecular marker and genomics allow conservationists to identify genetically important plant population more accurately. Instead of relying only on visible traits, which can sometimes be misleading, conservation efforts can focus on populations that truly have high genetic conservation value. Secondly, these genomic tools improve efficiently. Large amount of genetic data can be analyzed in a shorter time, which speeds up the process of assessing genetic diversity and identifying endangered species. As a result, conservation action can be planned and implemented more quickly and effectively. Finally, the use of genetic information increases the success rate of restoration and reintroduction programs. By selecting genetically suitable source population, restored plant population show higher survival rates, better adaptability, and improve long-term population stability.
In conclusion, conservation challenge faced by plant species due to limited genomic resources can lead to rapid biodiversity loss. However, this challenge can be addressed through the use of genomic techniques and molecular markers. Through this approach, biotechnology provides significant value and impact in supporting biodiversity conservation in a more efficient and effective manner.